Okay, so believe it or not, here we are, the final lecture of the final chapter. The course is almost done. Hopefully you've enjoyed it as much as I have, and hopefully you've been doing well, you've been understanding the homework sets and the exams, and things are going very well for you. More importantly, I hope you've acquired the skills and understanding that you need from this course for whatever future goals you might have. So here in our final lecture, we're going to discuss viruses, those pesky little things that make even the basic thing like the definition of life very difficult, because viruses can exist for quite a while in a state that we wouldn't associate with any of the functions of life, and then suddenly they spring back to life. So these viruses have caused trouble for quite some time, putting a kink in the works for the biologist to do something even so simple as define life, the whole study of life being biology. Okay, so viruses are small particles of DNA or RNA that require a host cell to replicate. So they can't live on their own, which calls into question whether or not a virus is truly alive. It's able to take over the machinery of a host cell, but it can't do the functions of life on its own. So it's a very difficult thing. These viruses sort of defy the odds in terms of what we've come to understand for the other types of life. They can cause a viral infection when that DNA or RNA enters a host cell. They are synthesized in the host cell from the viral RNA produced by viral DNA. And then vaccines are inactive forms of these viruses that boost the immune response. So they help us promote production of antibodies without ever really being infected by the active virus. So by having those inactive forms injected is the usual method of introducing those vaccines, our immune system gets to know the virus RNA or DNA and therefore build up all these antibodies so that should we be infected in the future, we've already got our armies ready and we don't succumb to the viral infection as we would if we had not received the vaccine. And so here we see a cartoon of that process. So we see that sort of sci-fi looking virus there doing its thing, attaching to the cell and injecting its DNA into the infected cell. And then once the virus gets its DNA in there, it hijacks our normal cell machinery to form many, many replicates of its DNA, and then that cell can burst, and those new viral DNA copies can go out and infect uninfected cells. Okay, so here's the problem with viruses. The types of disease caused by viral infection, even though we don't have terribly many diseases listed here, and this is just a small selection of diseases caused by virus, right? It's just select diseases. If we look at the virus, even for something like the common cold, if it's a coronavirus, there's over 100 types. If it's a rhinovirus, there's even more. So that's why you can, if you're like me, end up with common colds, at least one, if not two or three every winter, because although your body does develop immunity to that particular strain, there's, again, the other types that you're subject to. So that's what makes viruses so interesting and so overpowering at times, because there are so many different types, and the body may learn how to fight one type and then be infected by another type, and you're right back at the common cold. For influenza, there's not so many types listed here, but certainly people do get the flu vaccine and still end up with the flu because the producers of the vaccine guessed wrong, or you happen to get a different strain. So even though we don't say there's multiple types, there are multiple types there as well. Looking at warts or herpes or HPV, lots of different things there. Leukemia can be caused, or other cancers, and AIDS, acquired immunodeficiency system. Those are all possibly caused by a retrovirus. AIDS is definitely caused by a retrovirus. So that's an instance where we talked about viruses as being possible causes of genetic disorders a couple lectures back, and there's the cancers that this particular types of cancers anyway. Leukemia, for instance, can be caused by a retrovirus. we've got hepatitis, and then the different strains, Hep A, Hep B, Hep C, mumps, mononucleosis, chickenpox. A lot of these we have effective vaccines for, but not always. And certainly with a coronavirus or rhinovirus, you can't give people 210 vaccines. And that's not even to say that the particular types you're inoculated against won't undergo alteration and become forms that your body can't fight effectively. So viruses are small but powerful. So on the previous slide, we did have the retrovirus there for things like leukemia, cancers, AIDS. So a reverse transcription, a retrovirus which contains viral RNA but no viral DNA enters a cell. That viral RNA uses reverse transcriptase to produce a viral DNA strand. So this retrovirus that didn't have its own DNA actually uses your cells, if it infects you, to produce the viral DNA. And then that viral DNA strand can form a complementary DNA strand, and those new DNA strands use the nucleotides and enzymes in the host to synthesize new virus particles. So really, really interesting and very deadly in the case of AIDS, for instance, or those cancers as well, potentially deadly, the work of these retroviruses. And here we see a cartoon of that reverse transcription. So the virus infects the cell, uses that reverse transcriptase, which fortunately we've been able to find some inhibitors like AZT and DDI. These are things that 30 years ago we didn't have in our arsenal, and unfortunately HIV almost always progressed to AIDS, and it was typically a death sentence for anyone infected. Fortunately, with things like AZT and other inhibitors, we've been able to essentially halt the progression of AIDS or HIV into AIDS, and people are living much longer, and that's a very, very good, important fight in the battle against the retroviruses. So you see there, they can, if we don't have the inhibitors, we have the viral RNA using reverse transcriptase to produce viral DNA, and then you get a provirus, and so on and so forth, and it's ready to go out and make new viruses to infect new cells or new organisms. So we've already talked about it a little bit, but let's take some time to thoroughly explore HIV and AIDS. So the HIV-1 virus is a retrovirus that infects T4 lymphocyte cells. So that decreases the T4 level, makes the immune system unable to destroy harmful organisms, and it's associated with an increased chance of developing pneumonia and skin cancers associated with AIDS. So it's not the virus that was killing the AIDS patients back before we had AZT and other drugs in our arsenals. And unfortunately, still people do die. I'm not here to say that it's always a success, but AZT at least allows us to prolong the lives of most people stricken with HIV-1 virus. But those who eventually succumb to AIDS, acquired immunodeficiency syndrome, they die because their immune system's not functioning correctly, and they're developing these disorders like pneumonia and skin cancer that they would not have otherwise developed probably, and they can't fight because of their T4 levels being nearly nil. Okay, so fortunately, as I said, we do have AIDS treatments these days, although certain areas of the world don't have adequate access to them. Regions in Africa is where there's still very high incidence of death from AIDS because there aren't readily available altered nucleosides like this AZT that we mentioned, or DDI. In areas where that's not available, there are still many deaths, but in more of the world, we've been able to get these AIDS treatments that prevent that reverse transcription of the viral DNA, and then those, when they get incorporated into the viral DNA, the virus is unable to replicate. So it sort of halts it in its track, although it is something that you're typically on for the rest of your life. So it's not a single dose cure, it's a long-term halting of the progression of the virus. All right, so here we see some of these structures. So AZT, as it's commonly known, is actually 3' azeto, which deals with the nitrogen there, N3 group off of that carbon on the sugar, the pentose ring. So 3' azido, 3' deoxythymidine, so that's AZT. Another DDI there is 2', 3' dideoxyinosine, and we've got some other ones down below, but these are some typical AIDS treatments that are available. And again, they do have side effects, and they are typically prescribed for the long term, so patients do have to deal with those side effects for quite some time. So in addition to those compounds we just mentioned, there are other types of AIDS treatments that involve protease inhibitors, so such as saquinavir, Inverase, indinavir, Crixivan, fosamprenavir, Lexiva is a little easier to say, not for me apparently, and ritonavir, Norvir as it's more commonly known. So these protease inhibitors modify the active sites of the protease enzyme, which prevents the synthesis of the viral protein. So another way to attack the problem, and again, another set of HIV or AIDS treatments. And so I was smart enough just to put that Lexiva name here, but here's its structure, and you see some sulfur oxygen bonds there. It's double bonds. We've got amines, benzene rings, technically, and aniline down there at the bottom. Lots of interesting things. There's a phosphate group, so you can see where it would fit into a lot of what we've discussed in terms of organic functional groups and biological compounds. So this is one that would be a protease inhibitor. Before we end our final lecture for the course, let's go ahead and have one last learning check. So in this learning check, you're asked to match the following terms to their definition. So 1, 2, 3, 4, match up with A, B, C, D. Stop the video here while you do that. Start us back up when you're ready to check your work. Good luck. Okay, and there we have it. So A, a virus containing RNA, is 2, a retrovirus. B, small particles requiring host cells to replicate. That's our broad definition of a virus, number 1. C, a substance that prevents the synthesis of viral proteins. That's 3, a protease inhibitor. And then 4 is D, using viral RNA to synthesize viral DNA is called reverse transcriptase, and that's what our retroviruses do. So there we have it. Hopefully you were able to get those correct, and you're understanding viruses and really all the content of the course at this point. But certainly there's still a little time left. If you are having trouble, please do reach out to me for some help so that we can get things squared away before that last exam. It's been a pleasure. I hope you've enjoyed the course as much as I have, and certainly I hope the best for you personally and professionally.